we'll start with implantation proper. What happens, how exactly implantation takes place, and what are the changes happening to the blastocyst at this point. The second week of development, okay?
Implantation of the blastocyst usually occurs 6 to 8 days after fertilization.
Remember
By the end of day 8, the blastocyst has burrowed into the endometrium of the uterus. At this time, it is composed of two main components: the outer cell mass, the trophoblast, and the inner cell mass, the embryoblast.
Okay, so now we're just differentiating into two crude terminologies, that when it's burrowing inside, the outer one is the trophoblast and the inner one is the embryoblast. Okay, so mostly from outer cells, the trophoblast, the placenta will form, and from the inside embryoblast, the embryo proper is going to form, right? Now, as far as this trophoblast layer is concerned, it's going to impinge upon, form the placenta. But the dynamism of embryo is far more as compared to that of placenta. So I have included two slides to tell you what happens, what are the changes going on in the placenta, but that's not embryology. But still you need to know what and how this placental development takes place. That I'm going to discuss towards the end of the lecture. Right now, try to focus on what are the changes happening between these trophoblast and embryoblast cells, because right now everything looks very confluent, almost similar. Up till now, the changes that are taking place in the entire, the blastocyst. But now slowly the differentiation is starting from this point in time.
As the trophoblast makes contact with the endometrium, it differentiates into two layers.
Now the trophoblasts are also differentiating into two layers, the syncytio and the cyto.
An inner cytotrophoblast and an outer syncytiotrophoblast. The embryoblast differentiates into a bilaminar embryonic disc.
Now, even the embryoblast is differentiated into two germ layers. Okay, one is the hypoblast cells, which are the more cuboidal yellow-colored cells, and the epiblast layer, which is more of columnar and the blue-colored cells. These are the epiblasts.
Composed of two cell layers, the hypoblast and the epiblast. Soon, an embryonic disc has formed. A cavity begins to appear between the epiblast and the cytotrophoblast, known as the amniotic cavity. Cells originating from the hypoblast begin to migrate, forming a thin membrane, which covers the inner surface of the cytotrophoblast. This is called the exocoelomic membrane.
So now what has happened is that these hypoblast cells slowly they start, lining the cytotrophoblast layer, okay, and they form a cavity within. Obviously, if anything is to just line a hollow space, a cavity is formed, right? And this cavity is called as the exocoelomic cavity, and this is called as the exocoelomic membrane. Or it's called as the primitive yolk sac earlier on. It's also called as exocoelomic cavity because it's surrounded by an exocoelomic membrane.
The exocoelomic membrane and cells of the hypoblast together form the walls of the primitive yolk sac.
Yeah, you can call it primitive
By
yolk
day
sac.
9, the blastocyst is completely embedded in the uterus wall. At this stage of development, the growth of the syncytiotrophoblast and cytotrophoblast is much quicker than the bilaminar embryonic disc.
So can you see that the syncytiotrophoblast, now what exactly syncytiotrophoblast is? That a lot of mass of cells, which is being, propagated inside by a lot of proteolytic enzymes being secreted by these, cytotrophoblast, these pink colored cells. They are the ones which are actually causing, can you imagine burrowing inside, that too a syncytium of cells is burrowing inside, okay? So it must be having something to burrow, So it keeps these cytotrophoblast is secreting proteolytic enzymes, and these, syncytiotrophoblast are finding a way to go inside the endometrial cavity or endometrium proper. Endometrial cavity, they've already eroded. Now they're going inside the endometrium. And now how they're going? By the secretion of these proteolytic enzymes. But at this point in time, you're seeing that there's a rapid growth of cytotrophoblast and syncytiotrophoblast. And the embryo proper, this disc, that's not growing with that same speed.
Small holes called lacunae begin to form in the syncytiotrophoblast as it continues to expand. By day 12, the lacunae stop growing and fuse to form large interconnecting spaces called lacunar networks.
This I'll explain you when we'll be dealing with the placental development.
Capillaries in the endometrium surrounding the developing embryo dilate, forming maternal sinusoids. As the syncytial trophoblast continues to expand, enzymes begin to erode the lining of the sinusoids, and uterine glands, allowing maternal blood and uterine secretions to flow into the lacunar networks, establishing a uteroplacental circulation. The blood and uterine secretions only come into close proximity to the embryo, allowing the exchange of gases and metabolites. Around
So this I'll be explaining you when we're dealing with the placental development.
the same time, a new population of cells appear between the inner surface of the cytotrophoblast and the outer surface of the primitive yolk sac.
Are you getting this? Can you see this crimson color cells which have now kind of inhabited this area which was there between the cytotrophoblast, the pink one, and the hypoblast cells, or let's call it the primitive yolk sac now, the primitive yolk sac. So there's a whole population of cells which is like kind of sitting in between this whatever area it was, between the primitive yolk sac and the cytotrophoblast. So let's see what we call that.
Known as the extraembryonic mesoderm.
Now this is mesoderm, extraembryonic mesoderm. This is the embryo proper. So whenever we'll have a mesoderm, it will be somewhere in between these two germ layers, right? But this one, this part of mesoderm is the extraembryonic mesoderm, remember, because it's outside the embryo proper.
Large cavities begin to appear in the extraembryonic mesoderm. These gradually fuse to form one single cavity, called the chorionic cavity.
Now this again, it forms a cavity. Now it's called as the chorionic cavity. So here we have the amniotic cavity, the blue one. Here we have the chorionic cavity. So now you're trying to see now slowly the things are getting to the present day, fetus or embryo that you know of. So slowly these changes are kind of making more sense.
Around 13 days after fertilization, a large portion of the exocoelomic cavity is pinched off, forming a smaller cavity, the secondary yolk sac.
So now part of it is like pinched off and it's gone. And now this primitive yolk sac becomes a secondary yolk sac. It is a yolk sac, true, but it's now the secondary yolk
By
sac.
the end of the second week of development, the chorionic cavity enlarges, and the bilaminar embryonic disc is joined to the trophoblast by a band of extraembryonic mesoderm called the connecting stalk, the future umbilical cord.
This will be the future umbilical. So roughly now you're understanding how things are happening. This is a chorionic cavity. This is the amniotic cavity. From it, this is going to be formed the embryo proper. This will be the umbilical cord. Okay? Things are now making some sense. So now we go into the second week of development, in which we'll be talking about the formation of the 3 germ layers, which is why I am actually taking this lecture up. Because once I leave you to the development of the 3 germ layers, it's from there I'll be taking up almost all my lectures to come. So we'll be discussing about the CNS development, the GI development, the urogenital development, and these systems are important for us gynecologists, not the CNS so much, but the GI system, the GI tract, and the urogenital system, because we work in the proximity of all these structures. So for us to know these things, the embryological origin is very, very important, because germ lines usually is the one in which, metastasis takes place. Even anatomical structures, even the origin of the arteries, sometimes the nerve supply is all based on the embryological origin. So always important for you to know the basics of what you're dealing with. So I will be touching upon the, the circulatory system or the CNS, but I'll be taking more in-depth analysis of GI tract and of course urogenital system. It's always asked in your exam. So we'll be discussing with that as well. So now I'll take up these slides quickly. So the 30 hours post-fertilization, the mitotic divisions, they start. And 357, remember this, 357, morula, blastocyst, implantation. Morula, blastocyst, implantation. Okay. And over here, you can see roughly the corresponding changes, how it looks like. So, okay. Then we talk about day 7, 8, 9. Remember this day 7, 8, 9, bilaminar germ layer has come. Amniotic cavity starts to appear. primary yolk sac has now fashioned up. Okay, so day 7, 8, 9. I usually remember this as days 3, 5, 7. Okay, you know, odd numbers 3, 5, 7, morula, blastocyst, implantation, and then from 7 only 7, 8, 9. Bilemina germ layer, amniotic cavity, primary yolk sac, and then just jump by 3, 12, 13, 14. So you have the extra embryonic coelom. Now everything is making sense. Okay, extra embryonic coelom we've just dealt with, right? Extra embryonic mesoderm comes up. Day 13 you have implantation is almost complete. Chorionic cavity starts appearing, and of course this primary chorionic villi starts to impinge upon. And by day 14 here comes the applied embryology. HCG production starts from day 14, from the day of fertilization. So the day you have a missed period, if you try to do the UPT test because of this HCG production, it comes out to be weak positive, and then slowly it starts getting more and more darker. The lines become more and more dark.